All right. Well, I just want to welcome everyone here today to our COVID Force Talk. We're really delighted to have Dr. John Roback with us today. So I'm Stacy Heilman. I'm the Associate Vice Chair of Research and Department of Pediatrics, and I am an active fan and member of the RADx team. I've actually known Dr. Roback since I first arrived back in 1996 as I became fast friends with his wife. So it really delights me that he accepted our invitation today to speak at COVID Force. Dr. Roback is currently a professor and executive vice chair for clinical operations in the Department of Pathology and Laboratory Medicine in the Emory University School of Medicine. He's also medical director for Emory Medical Laboratories. He received his undergraduate degree from Johns Hopkins and his MD and PhD degrees from the University of Chicago. His clinical work is in the areas of transfusion medicine, cell therapies, and hemophoresis. His research interests include understanding the metabolic changes that occur during red blood cell storage prior to transfusion and developing novel transfusion products. More recently, he has been deeply involved with developing testing methods for SARS-CoV-2. His research investigations have been generously supported over the years by NIH, other governmental agencies, including DoD, as well as private foundations and the National Blood Foundation. As you can imagine, he's perfectly positioned to be helping us tackle this crisis that we have on our hands. And so I'm really delighted to introduce him as he's about to tell us about asymptomatic surveillance for SARS-CoV-2, the Emory University Experience. Thank you, John.
Okay, well, thanks, Stacy and Barbara for the invitation. So I want to start with what I think is probably the most important slide in my entire deck, which is I just want to illustrate like all the people that were involved in this project, because I'm still worried that I forgot somebody. So if I did, I hope they'll forgive me. But I mean, this has only been going on for a year, but yet we had tremendous contributions from university leadership, including President Fenves and his team, and then from I've worked really closely with the team that was involved in the screening program. So Debbie, Rachel, and Josh had many conversations with Amir, Alex, and Sam as we were talking through how we were going to build this out. And then also sometimes it's forgotten about, but Kevin and Hillary in procurement were hugely responsible for us getting this up and running as fast as we did, making sure that we constantly had the supplies we needed. So that was a huge lift. As I'm going to show you toward the end of the presentation, really would not have been possible without Monica and the rest of her colleagues from the Office of Information Technology, used to be called LITS. But I mean, you'll see the heavy lift they did. We had pathology IT support. We really partnered with two companies, Quanterix and PerkinElmer, on bringing this up. And then our laboratory over in the Woodruff Memorial Building, which we call ELIAD, the Emory Children's Laboratory for Innovative Assay Development, all of these folks were involved at some level or another with this project, as well as other things we're doing over there. So I mean, just a huge slate of contributors, and none of this would have been possible without them. In terms of conflicts of interest, I don't have anything that's relevant to this presentation. So let me start with background. So during spring break of last year, March 2020, the Emory COVID Task Force instituted several measures to address the developing pandemic. So students were instructed not to return to campus, or if they didn't leave campus, during spring break, they were sent home. The remainder of the semester was conducted via Zoom, and then employees who could work remotely were directed to do so. And then while planning for what to do at the start of the semester last fall, it became clear that at one level or another, regular surveillance testing was going to be critical, especially prior to the availability of a vaccine. So Emory initially contracted with Peachtree Immediate Care in order to test faculty, staff, and students who were returning to campus prior to the fall semester. And then this kind of evolved then into a weekly screening program. They used a point of care assay from a company called Quidel. And then once again, about a year ago, in early August 2020, the task force started approaching several faculty to ask for advice on how we could insource the asymptomatic surveillance testing program. And when they came to me, I had a couple of experiences which I thought would really benefit the university in terms of this program. First, in my role as medical director for Emory Medical Labs, I was really acutely aware of the daily supply chain challenges with our molecular testing platforms for SARS-CoV-2. We try to do most of it on our Roche platform, but we also have some other platforms. And you know, during the spring and summer of 2020, there was almost a daily conversation about how we could get more test kits, you know, more disposables, that kind of thing. So it was really, there was a real crimp on the supply chain for molecular testing. And then I was also involved as a co-investigator on the NIH NIBIB RADx program that evaluated, well, to date, we've evaluated over 50 tests to detect SARS-CoV-2. And so kind of, you know, putting together, integrating that knowledge, I had proposed that I thought we had an opportunity to actually implement a very high performing but non-PCR assay for the campus surveillance testing. This was from a company called Quanterix. And one of the things I really liked about it was that we were at a point where we could do a contract and get, you know, basically guaranteed supply chain because we were approaching them before they'd really rolled out their tests in a big way. So their supply chain was really unencumbered, and we were able to do a contract with them, once again, through procurement that was really beneficial to us, guaranteeing us a certain number of tests, you know, per week. So that was my proposal, and that's what Emory decided to, you know, go forward with. I just wanted to show, I think, you know, most people on here are probably really familiar with the RADx program, Wilbur, Greg, and Oliver, you know, being the leadership team, and then, you know, Julie and Allie being really in charge of operations. And then there were a lot of different parts of this program. So, you know, there were collaborators outside of Emory. There was a biostats core, a regulatory core, engineering core, clinical studies core. And then this is kind of where I participated along with Nadia in my lab, and then Jess Ingersoll and Colleen Kraft's group. And where we were really participating was to build out panels to test these devices, then actually test the devices in a blinded way. So we had sort of a front seat to how these different tests worked. And this, Julie gave me this slide. This is just sort of a nice graphic to show, and this is not the complete list, but to show a subset of the tests from different companies that we've evaluated, along with some pictures. A lot of these tests are sort of point of care devices, but there are some that weren't. There are some that are central laboratory tests, including the one from Fluidigm, and then also the one from Quanterix that we ended up going forward with. So let me talk then about some of the decisions we had to make. So the first one was, and I've already kind of answered this, but you know, which testing platform to select. And we chose the Quanterix HDX platform, which runs something called the Simoa N antigen assay. And Simoa stands for single molecule analysis. So SIMOA, single molecule analysis. And I don't want to spend a whole lot of time going into the technology, although I think there's a couple parts of it that are you know, kind of cool. So when you think of your standard ELISA type format, What happens is, you end up using a relatively large volume of sample, about 100 microliters, and diffusion of the signal reduces sensitivity, and you really need to have millions of molecules there to be able to reach the detection limit. So what Quanterix did is they took sort of a different path to overcome that signal dilution. They really miniaturized their immunoassay. And so now they use a very tiny reaction volume. So they defeat diffusion, and you only need one molecule, so one, let's say, antigen interacting with an antibody to detect it. And you can see examples of that here. You have sort of this honeycomb pattern, and where you have a green, you can get a green signal there with one molecule binding to a bead. So let me show you on this next slide a little bit, sort of a slightly different view on this, but it's pretty cool the way it works. So this is where the reactions occur. And if this looks to some of you like a DVD or a Blu-ray disc, that's because these reaction discs, which are the same size, are actually made by the company that used to make all the Blu-ray discs for Sony. So it is basically the size of a Blu-ray disc. And it's got, and in that disc are etched all these micro wells that you can see here. They're showing them kind of on the side here, but they all look like these little wells. And the wells are of the size that one of these beads just fits in it, but two beads won't. So one bead will fit in it, and then, based on kind of the way, the stochastic way the reactions occur, you'll have some wells that don't have any beads, but essentially you have one bead per well. And here you have an example of what those beads look like. So those beads have antibodies to capture the antigen, and then they have a detection reagent here. And once again, as little as one antigen binding to the antibody, and with the detection reagent binding to that, will generate enough signal in these microwells to be able to detect it here. And that's what you can see. So it's a pretty cool, super sensitive technology. And it performed really, really well in our hands in RADx. So what I'm showing you here are data that we generated in RADx. So one of the things that we did to help Quanterix apply to the FDA and get EUA for their assay, their nucleocapsid antigen assay with the NP swabs, is that we did some, help them get samples and do paired testing. So what you can see are the predicate results generated in Emory Medical Labs using the Roche cobas. You can see the CT values. So basically how many cycles it took to detect that as positive. And then you can actually see the results on the Quanterix assay. So the fewer cycles, the more virus there on Roche, and then the higher viral antigen here. And you can see, pretty good performance, and really very good correlation down to a CT of around 33 or so. And interestingly, it's about 10,000 times more sensitive than the Quidel assay, which, as I mentioned, Peachtree Immediate Care was using. The assay's fully automated, and we were able to do a contract that prior to when they got their EUA, we were able to do a contract that sort of guaranteed us the essentially uninterrupted supplies of reagents. And this is a graphic we did a while ago, but sort of comparing some of the different assays that we tested as part of RADx. And you can see that in this case, we used a positive pool of patient samples that we diluted tenfold down to 1 to 10,000. And you can see that this assay really performed incredibly well, going to like our greatest dilution, and still showing up positive for this. So performed really well, especially compared to these other antigen assays out there. So really almost performs as sensitively as a molecular assay, but without having to really worry about the supply chain constraints. So, and then the other thing that people would want to know is, well, I mean, we've heard a lot about variants. Will this detect variants? And this is also data that RADx helped secure samples for that Quanterix would test. And at the time that this slide was generated, all of the variants that we had access to and shared with Quanterix were readily detected by their assay. And then here in this kind of greenish bar, breaking out the Delta variants. And you can see that the only one that was missed probably wasn't missed because it was a variant, but was missed because it had, you know, a relatively high CT, almost 33. And it's not surprising that, that this assay detects variants well, because the variants mostly have their, mostly the mutations and the variants are on the, you know, on the spike protein on the exterior of the virus. And this assay detects the nucleocapsid antigens. So where there are fewer, where there are fewer mutations. So performs really well with these variants of concern. So that answered, you know, kind of the first question, what platform would we use? And then the second question is what sample type? So Quanterix actually performs really well with NP swabs, anterior nares swabs, even plasma and serum and saliva. But what we chose to do in you know, consultation with the leadership team is to focus on saliva. And few reasons we decided to focus on that. First, you know, it's a pretty low impact sample type. And so early on when the screening program was rolled out, it was voluntary. So we wanted to have good compliance and participation, and the easier it was for students and staff to give a sample, the better we thought the compliance would be. So it's quite easy for you know, individuals to give about an ml of saliva. It's a pretty high throughput way to collect samples. Well, I'll show you what our collection site looks like, but you know, as compared to NP nasopharyngeal swabs, where you actually have to have a trained staff member collecting it, the saliva is all self-collected. So really high throughput and requires fewer staff. There was a potential to transition to sort of a sample drop-off model where individuals could collect, let's say, in their dorm room and drop it off. We never actually implemented that, but there was a potential for that. There's some data that suggests, and I'll show you that in a few slides, that at least with some assays, you know, the saliva could be was as sensitive and could even be more sensitive for asymptomatic testing. But the biggest problem that we really had to tackle with saliva is, of all the different sample types, it's the least homogeneous of the sample types. And so when we started testing saliva, you know, it was really challenging to test because, you know, frequently you'd get samples that were way too mucoid to even be able to pipette. But fortunately, we were, you know, we were getting some good advice from Georgia Tech, and for their asymptomatic surveillance testing, where they also use saliva, they ended up using a little device that looks like this called a bead ruptor, which they actually called it their like secret sauce to make their program work. And essentially you have a small tube that the saliva is collected in, and that tube has very small ceramic beads in it. And then those tubes go into this device, which shakes them really vigorously. And so the beads mechanically disrupt the sample, break those mucus threads, and now it's easy to pipette. We did have to modify their program a little bit because they used 6 beads in 30 seconds in this device, and that really destroyed the antigen reactivity. But we titrated it down and came up with a protocol that actually didn't negatively impact antigenicity and was much easier to pipette, not only pipetting manually, but doing robotic pipetting, which I'll show you some slides about that later on. So then the third question that we had to face is, how do we manage low positive samples? You know, we wanted a sensitive screening system. And I showed you that the saliva or the, I showed you that the Quanterix was quite sensitive, but you know, every individual that goes into the screening program, let's say the students, if they're asymptomatic, but they screen positive, then, you know, they get kind of whisked away to the conference center where they're, where they're, you know, socially distancing for, you know, over a week. So we wanted to make sure that we weren't sending people over to the conference center who might have, you know, a false positive result. So how did we address that? And so what we ended up doing was implementing a screen confirm method, which is really similar to how blood donor screening is done. So you screen with a really very sensitive assay, knowing that some of the positives are going to be false positives, but then you take all of those positives and follow up with a very high specificity confirmatory test. So any sample that's positive on both, then you have, you know, high confidence that it's a true positive. And we ended up using an RT-PCR assay from Yale called the Yale Saliva Direct Assay for this. One interesting feature of this program is that because we're using Quanterix, the Quanterix antigen assay to screen and the Yale Saliva Direct to confirm, not only are we confident that any individual who has a positive result on both, you know, does have an infection, but we have one test that's detecting antigen and another test that's detecting viral RNA. So that really raises our confidence even more that, you know, we have individuals here who have both the viral antigen and the RNA. Even though they're asymptomatic, they've got, you know, they've got the virus. These are the sort of individuals that should be isolated in the conference center. So so we ended up using the Yale Saliva Direct. We looked at a couple other assays, but Yale has one of the reasons we went with their assay is they had a really interesting relationship with the FDA. When they initially put in their EUA application, they essentially said to the FDA, we not only want, you know, we not only want this EUA, but we would like you to allow us to qualify other labs around the country to use this assay so we can really disperse testing. And so their mandate really was not just to use it at Yale, but to allow as many other labs as possible to jump on that EUA, which is what we were able to do. This is a paper that they published, you know, almost a year ago on their assay in the New England Journal. And one thing that they showed was that when looking at matched samples, and these were in asymptomatic healthcare workers, which is important because we were using it for asymptomatic, and they're asymptomatic healthcare workers, if they compared paired samples between those healthcare workers that collected a nasopharyngeal swab and saliva, you can see for these almost 10, I think it was 8 samples here. For these 8 samples, these paired samples, there was only one example where there was a higher signal in the nasopharyngeal swab and lower in the saliva. And the rest of the 7, they actually got a higher signal in saliva than the nasopharyngeal swab, which gave us some comfort that that this would be, you know, this is a good confirmatory assay for asymptomatics. And then they also showed how looking at the internal control on the assay, which is the RNase-P, they showed how if healthcare workers self-collected sort of an anterior nares swab, they said it's an NP swab, but I don't like getting an NP swab when somebody else collects it, and I'm kind of dubious that these healthcare workers were self-collecting an NP swab. It was probably more like a mid-turbinate, but you can see that compared to that swab, saliva actually had reproducibly higher levels of this internal RNase-P as a indicator of sample quality. So really the Saliva Direct looked like a very good assay for us to use for confirmatory testing.
So can I interrupt just for a second? So this is really fascinating. And it occurs to me, I wonder how important is timing on this in terms of when the exposure was? Is that a factor as well? Because this data looks really clean, and I have to imagine there's some kind of curve about when it's most sensitive and specific.
Yeah, yeah, and that's a great question. You know, the issue with the Yale Saliva Direct is that I'm not actually that familiar with any data they've done or any data they've generated on symptomatic testing, which is how you'd probably get the best assessment of how long after the onset of infection the screening is. So, but that's a very good question. This got us up to about the end of 2020, and and you know, there a lot of work happened that I'm not showing you right now, but around the end of 2020 in November, we actually took delivery, installation, and then validated 2 of these HDX instruments. So we're showing in this lab in WMB, we're showing the 2 HDX instruments right here. You know, there were I always, you know, told people that if we were bringing up a new test in Emory Medical Labs, this is not the way we would do it. We wouldn't be doing, you know, 7 things in parallel. We do it all in sequence. We do one thing, and then we do the second, and then we do the third. But, you know, we had to kind of do everything in parallel to get up and running and be able to start, you know, testing before the end of the year to take over testing. So in parallel with, you know, purchasing, installing, and validating the instruments, we were also hiring staff. But for that first week of testing, actually, there were only 3 of us who were actually qualified to run the test. There's, you know, Andy Nish, who's down here in the hood, actually pipetting the samples. And then there's you know, Hans Verkerke, who's an MD PhD student who was helping us. And then there's me actually like kind of taking this selfie and showing the 3 of us in the picture. So for the first week of testing, it was the 3 of us doing it, because we hadn't gotten any of our staff in yet. And then fortunately, the next week, starting November 23rd, one of our medical technologists, Bill Osick, started. Here's Bill. And then the week later, our other medical technologist, Caleb McClendon, started November 30th. And here's Caleb. And so we're really happy once we got to the week of November 23rd, because we could put this in the hands of 2 people that were, you know, way more qualified to do all the hands-on testing than we were. But it all started with the 3 of us actually doing the testing. And then, you know, also in parallel, so you know, Josh and Debbie and Rachel were working out the protocol for, you know, sample collection and pickup and delivery over to the laboratory. And this kind of shows you, you know, we really started very modestly in November. That is, we had, you know, we had pickups at the conference center and at Woodpeck, and then also we had a couple pickups each day at Oxford. So we only had a couple different sites and relatively small numbers of samples. And then those would get delivered to the lab. So the lab is over here. It shows when the samples would be dropped off, when we put them on the instrument. So even though when we started doing 1,000 samples a day seemed like a lot, it you know, kind of compared to what we ended up building to was you know, pretty modest actually. But and you know, Josh and the team worked through a lot of issues once again in parallel with, you know, the optimal courier schedule. You know, what are the optimal times for the collection sites? You know, when should the samples be delivered? Do we want to run them all the day we receive them? Could we hold some over, run them first thing in the morning? And then that involved a lot of discussions, you know, with student health and student leaders about, well, I mean, what should the turnaround time be? Should we inform everybody within 24 hours? What about 48 hours? So a lot of discussions about, you know, kind of the constraints on the system and what the best way to run it is. And so when we started that first week, just, you know, Andy, Hans, and myself, we were running, you know, 5 days that week, Monday through Friday. We were doing, you know, started with around, I think the first day actually was less than 150 samples. By the end of the week, you know, we did 500 samples. And then we actually, those next couple weeks between after Thanksgiving to right after the first of the year, when most students were gone from campus, we were only operating 1 to 2 days a week, about 250 samples a day. And that helped us get the additional staff in, train everybody up, and then be ready for the beginning of January, where we went, to 5 days a week. And at that time, our goal was to be able to test 1,000 samples a day. So, so then and that was working pretty well. And then our next decision was, how are we going to address sort of our progressively increasing testing goals? Because at the beginning of, January, the January, February timeframe, nobody had a good sense at all of how the pandemic was going to progress. And nobody knew. I mean, did we have to say that absolutely everybody on campus, who even sets foot on campus, has to be tested twice a week? Were we gonna use a different testing cadence? Were we gonna prioritize testing? It just wasn't known. So we decided to, set sort of a goal that that if we achieved it, meant that however the university wanted to organize testing, we'd be able to test that many samples. So that goal became 30,000 samples per week. And that required us to do two things to meet it. First, we had to expand staffing, and then we also had to introduce sample pooling. So on this slide, I have a couple pictures. This is when President Fenves was actually visiting the screening lab. And this is, just to show you, here's Bill showing, showing the president, the Quanterix operation. Here we have a couple of our staff going through the confirmatory testing with the saliva direct. Here we took a picture with President, out in the quad, and this shows a number of our other, team members here. And just to, reemphasize again, like all the people who were involved with this at various levels, but about half of these people on this slide are actually like the hands-on people that are doing the testing day in and day out. And I'm showing you then the robotic pipetter that we implemented, so we could then implement pooling. So, and so we did a lot of manual validation of pooling, and we initially validated it so we could take 3 samples, for example, the ones in these blue wells here in a plate, like column 1, rows A, B, and C, and put them into one pool. And then we did a lot of work manually looking at different pooling sizes and different parameters, just to confirm that we were basically getting the signal, or approximately the signal we were expecting when we did pooling. And then once we were comfortable with that, we had to automate it. Now, before I really go into, how we automated it, I thought this would be a really good time to switch gears a little bit and tell you about the IT component of this, because IT was actually huge. Like, none of this would have happened without, the IT input we had, from Monica and about a dozen of her, colleagues that, for the last year were helping us do this. And planning started 8/20. They started the IT build last October. I showed you where we opened 3 collection sites, 2 analyzers, up to about 500 samples actually per day, up to about 500 samples per day that first week, and then 500 samples per week between Thanksgiving and Christmas. And then last January went to 5 sites, 5 to 8,000 samples per week, implemented a couple additional analyzers. And then actually our big expansion occurred in April, where we now not only collected from 6 sites, but we had robotic pooling. We really made a more robust data pipeline, and could do up to about 15,000 per week. And then sort of our ongoing improvements, so that if need be, we can test up to 30,000 samples per week. And, just once again, laying out all the different partners we had, to make this happen. I've talked about some of them. I haven't talked about, groups like Campus Life and the provost that helped us work through sort of what different models looked like and did they meet the needs of the of the students and staff. And really the IT skill sets. I mean, we had lab informatics and analysts, system administrators, software engineers, et cetera. So it was really, I mean, it was like a huge IT lift to get this going. This just shows you sort of the overall IT workflow. And I know this is too small for many of you to see, but it considered what the testing program looked like from the perspective of, let's say, the student. How do they interface on their cell phones with the with the sample collection part of it? How do they do that? What is their what kind of reminders do they get? What kind of information do they get that that they have to produce when they show up to collect samples? And then how do they get the results? And I'll show you some examples of that. Then how about the folks that are working at the collection sites? How do they interface with the students? How do how much do they have to help the students? What kind of quality control do they have? And then a lot of it is, how the lab can track the samples. And then finally, the people that have oversight of the whole system, what they're able to see. So this workflow summary, and this is really kind of a superficial overview, but it tells you all the different data points where we had to do some kind of tracking. Now, in order to look at the IT components, I think it was kind of helpful to break it down into 6 different components. So, the student kind of, or the participant consent, symptom assessment, and then appointment scheduling, and then check-in to give a sample, tracking the samples, how we did everything in the lab, how we automated it, automated resulting in notifications, and then downstream reporting and dashboards. This just kind of shows like an infographic we have that goes over the entire testing program, where to go to get more information, what you do when you show up at the collection site, what to watch out for after your appointment. So the first part of this is the scheduling app, which the students and staff interfaced with on their cell phones. So one of the things they had to do was they had to review and sign a consent. Then they also have to go through a questionnaire. And the reason for that questionnaire is because this is the asymptomatic testing program. And if the students, let's say, had any sorts of symptoms, then that actually took them to a completely separate workflow, because then, let's say there were students, then they had to schedule to go to student health and get an NP swab. So just making sure that these were asymptomatic individuals. And then they actually got into the heart of the app, which was selecting the site and the date and time for their samples. So, here's an early screen saying you've selected the Emory Conference Center. Select your appointment date or change your location. So somebody wanted to select Monday, August 16th. And then they said, great, we have appointments on August 16th, select your appointment time. So they selected 9:20 a.m., which was available at that point. And then lastly, it generates an email that gives them all the details they'll need when they show up for their appointment. And here I've kind of, boxed out the specifics, but it's an email from, covidtest@emory.edu. It's to a participant whose email address is emory.edu. It's about their appointment confirmation. Thank you for scheduling. This is your name. This is your collection site. Here's the address. Here's the date. Here's the time. Here's your positive patient ID. This is your participant ID. Here's the little code that you're going to scan when you show up there. So it generates all that in an email, and so they have everything they need. Then when they show up at the collection site, what happens is that this is a largely touchless process. That is, they take that email and they scan their code. So what happens then, and I'm doing this very simplistically because I'm not an IT person, but essentially what it says to the system is, hey, you know, John Smith just showed up for his screening appointment. And then what John Smith, the student, will do is they reach into a bin that contains a packet in like a baggie that contains everything they need. They pick up that baggie and then they scan that as well. And that has a little barcode on it. So what that says to the system is John Smith, who scheduled his appointment for this time, picked up this baggie, and now the sample, which has the same barcode here, that result is going to be able to be tracked all the way to John Smith. So it's largely touchless. And so after they, after they pick up the bag, they scan it, everything is linked together. Then they walk over to the collection site. And here I'm showing the collection site at Woodpeck. Here's where they, picked up their baggie. Then they walk to one of these tables. And then there are instructions for how to collect the sample, but they basically take the barcoded tube out of the bag and then they generate their saliva sample. And then there are, kind of proctors that walk around, offer advice. And then after somebody's given their sample, one of the things we had to implement is that they walk around, with a little flashlight, just look at the sample to make sure that there's enough volume, because if there's not enough volume, we can't test. So they walk around, and then after it's all done, those samples get dropped off in a bin. So really, a ton of work just to get to the part where a student could come at the right time, give a saliva sample, and that saliva sample is in a tube with a barcode that gets linked to the student. So then the next part is the lab protocol automation. And this is something that we had right from the beginning that allowed us to track all the samples from Quanterix back to the student and generate an email with the results to the student. But this whole thing got, supersized when we had to go to automation. And this just, Monica put together a list of just a lot of the considerations necessary to help us automate what we were doing in the lab. And so, this list for many of you, probably you don't understand any better than I do, but I thought what would be helpful is that just to kind of show you graphically what happens. So this is the courier showing up to the lab with those tubes that were just collected, let's say in Woodpeck, overseen by, Rachel and Josh. So the courier shows up for the tubes, and the first thing we do is we put them in that bead ruptor so that they can be pipetted. But then this is when the IT takes over. So all the tubes get placed on the robotic pipetter, and that has a barcode reader. So it reads all the barcodes on all these tubes, which now allows us to track it back to the participant. And then what happens is that it automatically pipettes each one of these samples. It takes 3 of them together, and that's shown in blue, and pools them into one well for pooled testing. It also takes those 3 and pools them separately into another plate, a replicate plate, and that then allows us to throw away all these tubes afterwards, which really, when you're talking about doing up to say 6,000 samples a day, if you don't have to recap these tubes, that's a huge time saving. So it allows us to throw away those tubes, but still have an aliquot of that sample for confirmatory testing. Then what happens is that the software, after doing all of the barcode scanning and making the testing plate, it generates a CSV file which can be uploaded into the Quanterix instrument along with the testing plate. So now all the testing occurs, and then some of these samples which exceed a certain threshold, they're called presumptively positive. And after that happens, after we get a good run, then we just throw away the testing plate. And then that is fed back into the robotic pipetter. And what the robotic pipetter does is it takes this replicate plate, which has a replicate of each sample, and then it goes and cherry picks those replicates. So if these 3 samples here, when made into a pool, generated a presumptive positive result. What happens is that it goes back and cherry picks just these 3 to put them into a PCR plate for confirmatory testing. And then it goes ahead and makes one more replicate just of these presumptively positive. So those are the only ones we end up storing. And then we'll throw this plate away. The PCR plate goes into the Quant Studio to have the saliva, to run the, along with the reagents to run Saliva Direct. And then those results go directly into Open Specimen, where everything is managed by IT through Open Specimen. So these confirmatory results go into the logic to determine which individuals are confirmed positive. And we will throw those plates away, and then these storage plates go into the -80 in case we ever need to access those again. And so then, in Open Specimen, using these, these Python, and other scripts, I mean, once again, I think you're all getting the sense of the amount of work that IT had to do to make this work. But, it's doing all the work in the lab. It's doing a lot of checks all throughout in terms of things like, QC, make sure no samples are missing, et cetera, et cetera. It's tracking everything. And then presuming that everything, all the checks occur, then a notification goes out to the participants. So here's an example notification, email notification to somebody who had a positive test result. So once again, I blanked out who it's to, what their specimen ID was, et cetera. But it says, the result of your COVID test was positive. Please stay home, don't come to work or class, immediately isolate yourself. And, if you're on, if you don't have access to Clifton or Oxford, see your physician. But if you are on the Clifton campus, a provider from Student Health will contact you. So this is an example of a confirmed positive. Here's an example of a confirmed negative. Once again, blacked all this out, but the result of your COVID test was negative. Please continue to monitor for symptoms, including new or worsening. Here's a list of symptoms. Should any of these develop, self-isolate, contact Student Health, and then they'll later on get another email saying, hey, it's time for you to do your next asymptomatic screening. And then also, I told you that, there are issues with saliva samples, and a couple % of all samples, despite the safeguards we put in place, are just inadequate for testing. Either there's not enough volume to do the testing, or the sample is so mucoid that even by disrupting the sample using that bead ruptor, we still can't pipette it. So then they get this notification that they had a quantity not sufficient or an inadequate sample, and says, please schedule another appointment using the link here. So, we fortunately, it's only a couple of our samples, but that's, we still have a couple of those. So that's the participant notification. Now, there's a lot of other downstream reporting, which I'll show you in a second, but as I mentioned, we had to really maintain a very good chain of custody. So, make sure the person checked in and picked up the sample kit, sample was collected, sample was received in the lab, where that sample was in terms of, what batch got put on with the pipetter, what plate it was in, where it was in that plate for PCR, where the freezer location was, make sure all the interpretations are accurate, make sure the QC data, make sure the person was notified. And then lastly, update the dashboards, which I'll show you in a second. And this is the stuff that it's doing prospectively constantly with the IT infrastructure is doing, but it also supports a lot of questions. And, we realized as we started working on these that we had a lot of questions every day. For example, how many people have made appointments and how many samples do we think we're going to collect today? How many samples are in transit and when do we think they're going to get to the lab? How many samples do we think are going to have to be held over and then put on the analyzer first thing in the morning because they're going to arrive too late? How many positives did we have today? What are the populations, student or staff? Where, and then, from students, we'd get these requests if things got delayed. Where are my results? And that could be because, it could be because, the shipment was delayed. It could be because, let's say, one of our analyzers were down, and because we're doing preventative maintenance, so everything got delayed. But, or did the notifications get sent? And then we get a lot of these inquiries like, how long, what's our turnaround time from collection to result? We kept trying to improve that. And then sometimes, hey, we didn't have a sample. Did this person actually check in, or do they have an upcoming appointment? So the IT infrastructure really allowed us to do a lot of those queries. And then, just showing, some of the reports that we get where we can, track, the patient identifier, Emory ID, what the test results looked like, when the test was run, whether it actually went to Saliva Direct, et cetera. So, and then really more detailed dashboards that had a lot of aggregate data. So you can see that, this dashboard showed that there were 201 samples collected today, 935 this week. Total specimens tested since the beginning of the program, almost 120,000. So this gives you an idea. And these are the dashboards that really the team queries. But then there are these more generally available dashboards that divide up between the kind of confirmed cases here and then the Emory University screening. And you can see, and this is where our data would go, you can see that, when this data was pulled in the last 7 days, there were 7 students who came through the asymptomatic surveillance testing as positive, no staff and faculty. How about cumulative since July, since November 7th, 365 students, 37 faculty and staff, screening test capacity, et cetera. So really some of these really, great dashboards here that anybody can access to sort of see how things are going with asymptomatic surveillance. And then for my very last slide, and I'm happy to take questions, but I thought you might want to see some data about, how much testing we've done so far. So this is data current as of yesterday. So total samples collected over 120,000. That's composed of 3,500 distinct employees and 9,500 distinct students, and many of them giving repetitive serial samples based on the requirements of the testing program. We've reported out 118,000 distinct antigen results from Saliva Direct. The reason that this is less than 121,000 is that there are those rare inadequate specimens that don't actually get reported out as a result, are just, cannot test. We've done a total of 9,800 Saliva Direct RT-PCR results. Although one thing to point out to you is that that figure is affected by the fact that in order to keep turnaround times low, we decided that when we have one positive pool on Quanterix, that we will take all 3 of those samples and do Saliva Direct, even though at most we only expect one of those to be positive. But rather than retesting all 3 of them on Quanterix and then only doing Saliva Direct, we decided that this was the best way to keep turnaround time down. So 9,800 of those, 339 total positive results on Saliva Direct, 0.28% of the total. That's composed of 38 employees and 296 students. We had 5 people whose demographics we couldn't test, we couldn't identify. And then the flip side of the coin is how many confirmed positives, that is individuals who are either negative on Quanterix or on confirmatory testing, almost 118,000 or 99.7% of the total. And right now, even though we're all primed to do up to 6,000 a day, the most we've done, most Quanterix tests a day is 17,000 samples tested last April. But once again, if the university says we're gearing up and we want you to do 6,000 a day, we'll be ready for it. So that was my last slide, and it looks like we have about 5 minutes left for any questions.
Thank you so much, Dr. Roback. That was a whirlwind tour over what you've been up to for the last 18 months or so.
Right. It's been pretty crazy over the last year, that's for sure.
Yeah, we can see why you've been tired. You and the whole team. So I'd like to go ahead and invite people to either write in chat your questions or unmute yourselves and ask questions. I think if it's okay, I don't see anything in chat. I'll go ahead and start. I was really interested, especially as it relates to the ACME POCT, which is the parent grant for the RADx supplement. You know, we think about development of devices and what inventors need to think about. And so you're not the inventor in this case, but you're the adopter. And I was really interested in what you said about how you kind of got into the ground floor with Quanterix and didn't have to worry about supply chain issues. So I wondered 2 questions. I wondered, one, did you ever eventually encounter supply chain issues? And then my second question is, could you maybe talk about a few other factors that are sort of on the logistics business decision side about making a decision about technologies to adopt that might be useful to inventors?
Right. So actually with Quanterix, we never ended up with supply chain issues. We did have other supply chain issues though. So for example, with before we implemented robotic pipetting, when we did manual pipetting, we ran into some really serious issues one Friday with pipette tips, and we were using you know, I'm blanking on the name right now off the top of my head, but we were using a specialized multi-channel pipette that where the span between the tips could be adjusted, but only that manufacturer's tips worked, and we were like really short, and it wasn't clear we were going to be able to do all of next week's testing with the amount of tips we had on hand. So we did have problems with that, but never had any problems with the supply chain with Quanterix or with PerkinElmer once we implemented that. So that was a big deal. You know, one of the things that and I don't know if this will directly answer your question, but one of the things that I think that that any, you know, kind of budding medical device person could probably learn from our interaction with Quanterix is that from the very beginning, when we committed to Quanterix, we had at least one meeting a week, and sometimes a couple meetings a week with Quanterix. And it wasn't just the sales guy. In fact, like the salespeople kind of took they kind of took like, they kind of went to the back, you know. The people up front were all the way from the president, and I think I can show that on my next slide. So all the way from the president, you know, Kevin Krasovsky, and this is a, you know, company about 200 employees, you know, the president, Dawn Mattoon, who's the vice president for R&D. You know, she was on every call. We had Bev, who was the person who with her team was the ones that would come out, you know, get the device set up, do the preventive maintenance. These people, almost all of them were on the call every single week, and they understood that, you know, our success is their success, but also that really nobody had implemented their devices at this scale for surveillance testing. And so they thought, not only do they have to help us to get things up and running, but they also realized that, you know, whatever they learn from us, they're going to be able to, you know, translate to their other customers. So I think that, you know, if you have a customer who's like kind of ahead of the curve a little bit in terms of adopting it, it really benefits you to get your whole team together and have really regular discussions with that customer, because, you know, you can learn so much from their questions and they can learn so much from your questions.
Yeah, thank you so much. It's really about the clinical pull rather than the technology push. I think that's a really great lesson. So we are actually at time, but I'm going to ask you one more quick question. Dr. Shanazi wants to know, aside from going on the dashboard, what happens with the data on the positive as asymptomatic students or faculty?
Well, it's obviously it's used to identify those people who need to quarantine. It goes on the dashboard, and Emory University leadership is looking at that information a lot, especially to decide if we need to do some kind of a pivot in terms of our testing strategy. So it's used for that. And I know that there are, and I don't know how much progress we've made on this, but I know that there are some groups who are interested in accessing that data in a de-identified way to do some interesting sort of, epidemiologic studies. I'm just not sure how far that's gone yet, but that's certainly something that's a possibility.
Yeah, a treasure trove for sure.
Right.
Well, with that, I just want to thank you again and congratulate you and your entire team on such an impressive and important accomplishment. And thank you everyone for tuning in today to our COVID Force Talk, and we'll see you next time.
Okay. All right. Thank you.
All right.
Thank you.
Bye.